Welcome to the BioTalk Unzipped podcast, where we unzip the stories behind medical progress by sharing the latest and greatest advances in biopharmaceuticals and medical technologies in a fun, entertaining, and enlightening format. And now your co-hosts, Gregory Austin and Dr. Chad Briscoe.
Hi, everybody. Welcome to BioTalk Unzipped, where our mission is to unzip, unlock, and uncover the stories behind medical progress. I'm your host, Gregory Austin from Kansas City. I'm a life science professional in the quality and regulatory space at ECI. Along with me from North Carolina is my friend and co-host, Dr. Chad Briscoe. Hi, Chad. Chad is a bioanalytical scientific expert with Solarion, and we publish BioTalk Unzipped twice a month and bring experts from different corners of the pharmaceutical, biotech, and med device worlds to share their innovations, insights, career journeys, and personal stories to the show. Please be sure to subscribe, like, and comment on our episodes. Let us know what you'd like to hear about in the future, and thanks so much for listening. Today, we have on a good friend of ours from Ohio, Dr. Andy Vick. Andy received his PhD in pharmaceutical sciences from the Ohio State University, not an Ohio State University, just to be clear. Andy's worked in the field of toxicology, drug disposition, and PK/PD for over 20 years. He's worked at great sponsor companies like Biogen, Eli Lilly, Millipore, as well as analytical CROs such as Innovative, Will Research, and Charles River Labs. Andy is a past president of AAPS, an adjunct professor at Ohio State, and currently vice president at Attralus, head of toxicology, drug disposition, and PK/PD. Andy is a father, husband, missionary, teacher, and all-around great guy. Andy, thanks very much for being on the show today. How are you?
I'm doing very well, and it's a privilege to be here to join you both. I'm already realizing Chad and I could probably be doppelgangers, so we may get some comments about that, I'm sure. So,
yes, sir.
Well, it's a good thing we got your names in there, so we know who's who.
Absolutely.
So first off, BioTalk Unzipped would like to make a $100 donation to your favorite charity, Hope Ministries. And I did a little reading on it. Hope Ministries' mission is to preach the gospel, plant churches, train leaders, and aid them in planting, strengthening, and multiplying more churches throughout the Amazon region of northern Brazil, which is very cool. Andy, tell us a little bit more about the organization.
Sure, yeah, and it's there's a number of different charitable causes that I give to, both hopefully in time and talent and tithes. But the one that kind of just convicted me when you brought this up was, because we're thinking about our next trip already, is Hope Ministries and Brazil. The family, the Toth family, is kind of stationed in Castanhal, so we often fly into Belém, and we really have the awesome privilege of kind of going out on a tributary of the Amazon, which is like a short drive from Castanhal to Bujaru, and then we take a boat up and down river for a number of days, really to provide medical and to meet the medical, physical, and even the spiritual needs of some of the indigenous people along the river of some of the tributaries of the Amazon, which you can imagine are largely unreached. Both medically, spiritually, etc. And so I've been doing that trip. I think this will be coming up on my 5th trip, which we're already planning for next spring. Wow. And we'll take a medical team of 10 or 12, and I usually serve as the pharmacist. Now, I'm not a licensed pharmacist, but the country of Brazil thinks my credentials are sufficient to still practice pharmacy on the Guama River out in the Amazon, and it's a blessing. I hope I bless others, but whenever you're doing mission work, I think that you generally find that you end up receiving more than you're maybe even giving, and that's kind of the beauty of service. So
that's awesome. Well, thanks so much for sharing that.
Yeah, and Andy, I have to ask, because coincidentally, we just had a previous guest, Jonathan Hill, who also had a connection to Brazil, which was interesting. I also have a connection to Brazil. I was an exchange student there in high school, and I've been down there 3 times. Have some close friends, so I see this Brazil thing popping up, and I think a future episode, Gregory and I want to do I have a good friend who runs a pharma company in Brazil. So one of these days we'll have a specific Brazil episode. But,
awesome.
But yeah. So do you have a connection beyond the charity, or did the charity draw you to Brazil?
The charity is really what drew me to to Brazil, and it's been good too, because I always enjoy language even more than my language abilities are pretty self-limiting. I'm pretty decent in Spanish, but I kind of jumble Spanish together with Portuguese, and we survive. We also have some great translators, but a lot of the mission work I have done in the past has been in Russia and Belarus, but for obvious reasons, those countries are a little harder to get into, and will probably be for some time. So, but no, that was my connection to Brazil, is really the Toth family and Hope Ministry. So, but it's a beautiful country and very warm people, and sometimes those that have the least out in the out on the river communities have the biggest smiles and so much joy in their heart. That might be another conversation too, but it struck me at the moment just how joyful they are, those that have sometimes the least. Sometimes that other stuff just gets in the way.
That's truly phenomenal. Yeah, thank you.
And I think those folks can oftentimes be actually more faithful as well. I have a friend who leads an Amazon missionary group in Peru. Yeah. And same type of thing. They're in the Amazon. They don't have a lot of resources. They don't have the type of medical care that we have. And so when something happens, they rely on their faith first and then hope for an answer one way or another to get the help that they need. So it's pretty remarkable.
Absolutely. And appreciate
So, Andy, you're now with
Attralus.
Oh, yeah. Sorry, go ahead.
Nope. Attralus Therapeutics.
Thank you. I was going to ask, what is the right pronunciation? Because some of these are a little tricky. So Attralus, you're now with, it's an innovative company in clinical development, so looking primarily at treatments for systemic amyloidosis, if I'm saying that right, which is a rare disease in which a buildup of amyloid proteins occur in major organs, causing numerous issues in patients. Can you tell us a little bit more about the etiology of this disease and how Attralus is attacking that?
Sure, absolutely. So yes, just a little bit more about Attralus. Even in my own career, that's about 26 years now. They've kind of been bookended by organizations that have been 40 employees in size to maybe 40,000. Eli Lilly was the 40,000 at the time, and Attralus Therapeutics is the approximate 40, and so it is a startup biotech in every sense of the word, and they are focusing on biologic-based therapeutics and diagnostics related to amyloidosis. And our primary focus is systemic amyloidosis, so that toxic protein just kind of misfolds and accumulates in organs like the heart, liver, kidneys, spleen, etc. Probably the amyloid that we hear the most about is also amyloid in the CNS. So that's even an area of research for us as well, a little bit more preclinically. Systemic amyloidosis, we have our lead asset that's already in the clinic, kind of in a phase 1b2, and that's systemic amyloidosis. But again, that backup program, we know a lot about, A beta, alpha synuclein, tau, etc. And so this product, or this portfolio of products, at least on the ability to bind to amyloid, to opsonize it, to really render it detectable by the immune system, and then hopefully have the macrophages come up and do what they do best, which is just to clear things out, even including, hypothetically, the removal of this toxic amyloid, so that you can be not just silencing or not just delaying the progress, but potentially even clearing that bad actor out of the body and restoring normal physiologic function. So even systemic amyloidosis is, again, as you can imagine, when something that doesn't belong in a normal operating organ, like a toxic protein, it again really impacts the normal function of those organs, and it can in survival times that even rival some of the more aggressive cancer. So it is a life-threatening hematologic disorder, which was still an unmet medical, large unmet medical need, and privileged just to be doing our part to hopefully create some hope and what's a bit of a hopeless situation.
So Andy, what I understood is that you're sort of tagging the amyloids with a peptide, and then is it a macrophage comes along? I mean, the macrophages, I think of those as the Pac-Man of the Pac-Mans of the immune system, right? So is that how it's working? Did I understand that right? So they're coming along and chewing it up.
Absolutely. Okay. Our lead product is a ATO2, and you can find this on our website. That's public, and it does have a proprietary pan-amyloid reactive peptide sequence which is really, kind of, we feel like that the secret sauce, for lack of a better word, that has the particularly high affinity binding to various amyloid types, and is probably driving some of that not only that distribution but that target interaction. But yeah, what you said, Chad, is spot on.
Yeah.
Very good. And I just, for the sake of some of our non-scientific listeners out there, to break it down a little bit, can you briefly describe the role that toxicology serves in drug discovery and drug development?
Sure.
I know I open up a can of worms, but if you can just give us a quick synopsis.
We'll see how I answer, because my brain would probably answer this 10 different times. It's kind of like if you ask Chad, what does validation mean? It could mean 10 different things to 10 different people. But, as I evaluated, toxicology is really the science of I'll even step back. I'm going to answer a different question in response to this question. So pharmacology, I think we all generally understand. We're trying to evaluate the desirable attributes that a drug might cause on a body. What is its pharmacologic activity? What are those promising positive things that a drug might do to the body, whether that's a non-clinical animal species or whether that's a human animal. So toxicology is really just parallels that, but at toxicological relevance. So what are the adverse side effects that may be attributed? So not subtherapeutic, but kind of super therapeutic, even into that range where we start to really need to manage the side effects or the adverse effects. And so that often, unlike pharmacology studies, we're often exploring doses at multiples of what might be necessary to elicit pharmacologic activity. And we're doing that so that we can understand risk-benefit, often in a non-clinical environment. When I say non-clinical environment, that's generally rodents. And again, for most small molecules and large molecules, we're following ICH guidance. We're following other regulatory guidance, and there's, generally speaking, an expectation that we would do tox testing, not only in vitro, but also in vivo. Let's be realistic. The complexity of the human body is sometimes difficult to tease out and characterize in a test tube. So again, non-clinical studies, in vivo study, have their place. We perform those to the highest ethical standards, really under kind of a 3R mindset, so that we're minimizing the animals to the minimum necessary to answer the question of safety and tolerability. But circling back, that's generally a rodent species, which is mouse or rat. Often rat is the rodent species of choice because of its size, so it's a little bit more amenable to biomarker collection, exposure, and another endpoint collection. And then you have a large animal species, which is either a canine or a non-human primate. And that determines that, somewhat dependent on the biotransformation of your product, or maybe the homology to humans. So we will evaluate, again, the toxicity in those species in ways that really even not only mimic but support the clinical development. And we do that to understand risk and ultimately to minimize any unacceptable safety or tolerability of findings in a clinical outcome. So we really are protecting, we're protecting not only the animals, we're ultimately protecting the end user, the consumer, and that's, our mothers, fathers, sons, daughters, brothers, sisters, or even ourselves. And animals pay a incredible price so that we can manage clinical safety in the most respectful and ethical ways possible. That was a longer answer than I thought, but that kind of hits to it. And it's ultimately understanding risk benefit and also that once we're in the clinic, we have an understanding of managing some of the side effects or what to expect maybe in a clinic, so that we're not surprised. We never really want to be surprised in the clinic, but rather have that translational science be fairly seamless from preclinical or non-clinical environment to the clinical environment.
Excellent.
Yeah, well, that was a tremendous primer, actually. So I think and then what we take it from the animals to the first in man, and we still drop the lowest observable effect, like 1/100th or something
yes,
like that.
there is
dose
absolutely, depending on the indication and the risk benefit of that indication, but the 100x safety margin, and that's really designed to minimize surprises and to ensure safety in the clinical environment. Again, in indications that are much more life-threatening, where there may not be any therapeutic options, safety, there's perhaps a bit more flexibility with that same approach, but you're right, Chad, that 100x is pretty standard guidance. And that non-clinical work, there's quite a frenzy of it to start and support that first in human, our first human dose set of studies, but then we will run parallel to the clinical development, meaning that we'll need to extend the duration of those studies in animals. We may need to look at development of reproductive toxicity to support women of childbearing potential. And then in some instances, we may need to do truly lifetime exposure in rodent species and a chronic rodent bioassay to assess carcinogenic risk to make sure that we can understand, always kind of one step ahead of the clinical development, what the safety and implications could be to the human end users.
So if we were to take that back to your experience with Attralus, when you're dosing with these peptides, so in the tox phase, I'm curious, if you're looking at the toxicity of the peptides you're dosing, and then also, do you have animal models that there's probably animals that you can induce the amyloidosis in the animals, and so you're also looking at them almost as I'm going to call them patients, or diseased animals that would mimic the human disease.
Sure. So first of all, the ATO is a therapeutic antibody with that amyloid reactive peptide conjugated. So it is all one intact species that we're looking at, and so that is that therapeutic antibody with, I don't want to say antibody drug conjugate because not exactly the same context. That therapeutic antibody with a conjugated linker, aka ATO2, was evaluated not only in vitro but also in vivo and pilot dose rangefinders, which then led to I think our first set of toxicities, which is often the standard duration to support a chronic clinical candidate, is a 1 month repeat dosing. And we did a once weekly dosing because we at least want to match the dose regimen that's intended for the clinic, but maybe even more frequent to support what may be needed, a more frequent frequency in the clinic. And so we did normal rodents, and then we did non-human primates, which is often the case for biologics, especially therapeutic antibodies, because of pharmacologic relevance and homology to the end user. But your second question, that was a little bit difficult because, again, in a fairly rare disease, there's not a lot of great models. So we did have some models of amyloidosis where we very akin to even like a cancer xenograft, where we kind of transplanted an amyloidoma into a rodent model. And so we did certainly leverage those models, not only for an understanding kind of just frank safety and tolerability, also for biologic activity. And from that data, we can still extract that information. We could even use that model to even characterize what the exposure associated with pharmacologic activity was in that model, so that we can even do a human equivalent dose projection, just assuming, okay, what would be the equivalent amount of exposure that we'd need to see in a patient, with differences in clearance, with differences in just kinetics, to try to achieve the same, so that we could achieve that same exposure in patients and implied maybe the similar biologic activity. So on the animal model of disease, a little more difficult and kind of a rare disease state where you just don't have, it's not like diabetes where you have a number of different in vivo models and conditions to kind of evaluate the potential pharmacologic benefit. We had to stretch a little bit more to do that. And then, as you could imagine, back to our toxicology species, so those were normal animals. They weren't amyloid bearing, so in some regards they're not pharmacologically relevant per se. So if there was any target effects or target biology, normal animals aren't the best or the best means to evaluate that. So we just have to, in this business of science, particularly safety of science, we really do a good job of culminating and integrating all the data that we have access to, in vitro data, in vivo data, ex vivo data, sometimes even in silico data, to predict and manage clinical performance and risk as best we can. But that's a good question, because it even highlights there's no perfect scenario, and there's no perfect tools we really use. And I'm almost asking myself what's better, quality of data or quantity of data? Sometimes you don't have a lot of data, and so you better bloody be sure that the data that you do have is high quality, because you're going to be making decisions which can impact human health. And I take that, as we all do, very, very seriously. So.
Yeah, without a doubt. There's no easy drug development these days either,
No.
I don't think.
No.
I don't know if there ever was, right?
That's what keeps us young and keeps our full head of hair. So
exactly.
The challenge is maybe a little too cerebrally stimulating for me.
Stimulated it right out of the head.
Absolutely did.
So I'm curious now, we're kind of going in backwards order, talking about Attralus a little bit, but this is really interesting. What is there kind of a story with the company? These biotechs often have really fantastic stories. Is there something behind the history there, and how you landed there as well?
Well, sure. So I think the history really have to acknowledge one of our founders, Dr. John Wall. He kind of also serves as the chief scientific officer. He's affiliated with the University of Tennessee Medical Center. It's really his brainchild. I haven't even touched on the whole diagnostic as well, which maybe I will very briefly. So AT01 even uses some similar chemistry to bind to amyloid. And again, there's not great diagnostics for amyloidosis. That's part of the problem as well. But the AT01 binds with a label that could be I-124, could be technetium, that then you can image the binding affinity that product has, the AT01 product has for amyloidosis, so that you kind of have a visual indicator of the biodistribution of that product to the amyloid burden that a particular patient might have. And then not only could you potentially leverage that diagnostic for just the diagnosis of a disease, which is probably greatly underdiagnosed, but then even longitudinally, so that you could potentially see, combined with the therapeutic, you could you could measure improvement over time. Is that amyloid burden increasing? Is it decreasing? And so again, combining that diagnostic tool and with some of our therapeutic warheads could be a pretty powerful portfolio. So kind of back to the story again, Dr. John Wall was a founder. Spencer Guthrie, our chief operating officer, I think they really initiated the company that became Attralus. For the longest time, I don't know that I knew what Attralus even meant, but probably the two most prominent systemic amyloidosis is ATTR and AL amyloid and the U.S. So there you got we're all checking. Does that actually spell Attralus? It does. ATTR, AL, amyloidosis forms and within the United States is Attralus, and we've been operating, I think the organization, maybe that first conversation that may have occurred maybe 5 years ago, then has led to a good continued evolution where we've had received Series A and then Series B and a Series B extension most recently to support the ongoing clinical development. I had been on board for just a little over 2 years. I think, I joined in April 2 years ago, transitioned from 15 years on the contract research organization side back to kind of my humble beginnings on the innovator side. And we can certainly talk about that. I worried about that a little bit, but the transition was seamless. And I think, for a small company where you don't really have any labs, so you have to outsource everything. So I think the 15 years of outsourcing, the CRO experience served me well, and I was able to resharpen a little bit of the scientific blade. So I hope I answered all your questions there, Chad.
Yeah, but you did actually touch on one of the other questions I had,
Sure.
which was exactly thinking about that difference in working in biotech versus pharma versus CRO, and that you could probably spend a half an hour on this one also.
I could,
but I'm always fascinated because I've always been in a CRO, right?
I could. I'll try to hit this. Boy, I'll try to hit this at a high level. I would say that all of them have blessed me in a number of different ways, and all of them have contributed to who I am today, and that's that's still a work in progress. That's by no means anything that's close to being perfect, but I'd like to think I'm the result of a lot of the people that have been a part of my journey, and that's been also contract research organizations, startup biotechs, global pharmaceutical companies, and so I think, in my experience, they're all share more similarities than differences, and the similarities all generally are focused on a motivation to help others. Now, not always. Not always is there that. There's also a business acumen, and there's and the pharmaceutical industry is a business that needs to achieve certain financial objectives to stay in business, but there is always, across all those organizations, there was just a desire to help others, to improve the quality of life, to just as I said earlier, to help those that are struggling, to create hope in situations that might be hopeless, and I think that on the innovator side, which I'll come back to, that is just a little bit more finite. Meaning you have your own portfolio of compounds and this and that. Now on the CRO side, which perhaps I would say isn't for everybody, because I have kind of maybe jokingly referred to it as a dog year, but it's probably not that intense. But it is intense. But I'm a little bit of a fighter pilot DNA, and some of my most enjoyable experience have been in a CRO, because the only thing better than working for, kind of one boss, one company, where you really appreciate the culture, you appreciate the leadership, you appreciate the opportunity to kind of cook in the kitchen alongside a master chef, is to do that in an organization where you can now cook alongside 100 or 1,000 master chefs. Where that you don't just have one boss, but you have hundreds or thousands of bosses. And I had that. Now, that could be exasperating if you had a bad boss. Fortunately, I didn't have a lot of. I mostly had great bosses, and there's just an adrenaline. There's just if you're impact and you're influence driven and you like to create value in a CRO environment where you're doing that on a logarithmic scale. And I'll give Charles River a plug, where you're probably contributing to 85% in some way of the FDA approved drugs each year. That's hard not to get a little addicted to. So again, more similarities. I would say the scale and the pace and maybe the cultures and what is emphasized more in this organization versus another organization, throw in some diversity of life and of a San Francisco company versus, Indianapolis, Indiana versus Cambridge, Massachusetts. I mean, diversity comes in all sorts of shapes and sizes, which is a wonderful blessing as well. I chuckle a little bit, and I have to be careful here because I will say, even in my career, particularly when I was on the CRO side and I was supporting a number of startup companies and small, mid-tier biotechs, and they even often kind of fancied that the pace was just relentless. It was just this non-negotiable sense of urgency, and I would say relative to being an executive and officer at Charles River, I have found biotech to be a little bit of a Sunday drive. And I have to be careful there because, again, that might not be the situation for all small biotech companies. But and I enjoy working hard. I enjoy staying busy. And so even as I find in my current situation where I frankly have a little bit more life and the work-life balance, I'm just enjoying kind of using that time, not idly. I'm not a guy that can sit back. So, but like, getting back into the martial arts, and getting myself physically and mentally beat up, which I'll be doing again tonight, volunteering more at church than I was able to in the past, and just finding other ways to give back. Because again, the thing that I don't do well with, and now I'm deviating from your question, is I just don't do bored real well. So I like to stay busy so that I can keep creating impact, keep creating influence, and creating value, not only professionally, but even in our, personally in our own communities, our own backyard. So that, I completely went down a different rabbit hole, Chad, but I think I answered the heart of your question.
At some point in almost every podcast, I say this, and the way I frame up these podcasts is it's like we're sitting having a beer after a day at the conference, and so they tend to ramble around and go in different directions and bring different things up, just like you would if you're hanging out. And I love that you pulled out some really interesting things there. I've called CROs like being an ER doctor. I kind of like the fighter pilot analogy better, but they're both kind of just high stress, things thrown at you from left and right all the time. And I love that saying, I don't do bored well, because I don't either. And I think I'm going to use that with my wife, because she's always trying to get me to sit down.
Oh, boy.
When I sit down, I fall asleep. So
I agree.
That's funny. All right, Chad, I think it's time for our first segment, our favorite segment.
All right.
New news.
New news.
Glioblastoma vaccine turns brain tumors from cold to hot in humans and dogs is the title, published by Fierce Biotech by Helen Florsch on May 7th. So this novel mRNA vaccine shows promise against deadly brain cancer. Researchers from the University of Florida and a biotech company that spun off called Oncology developed a personalized mRNA-based cancer vaccine for glioblastoma. The most currently famous, of course, of the mRNA vaccines is for COVID, which we all know and love. Unlike traditional mRNA vaccines, this novel vaccine wraps the RNA specific to a patient's tumor in lipid nanoparticles called RNA lipid particle aggregates. These onion-like clusters activate a specific protein in stromal cells, stimulating a robust immune response against the cancer. So in early research, very early phase 1 trial, they've had 4 human patients, 10 pet dogs, all with brain cancer, showed promising survival outcomes. Of course, it's too early to understand the clinical effects in humans, but the patients are surviving longer than expected after the vaccine. The dogs indeed lived for another 139 days on average, far longer than the 30 to 60 day survival time that's normal. The vaccine's unique design holds potential for treating this aggressive cancer and other similar cancers. So exciting results, right? And I have a personal stake in this because my dad died of brain cancer many years ago. It's too early to get probably too excited because we don't have enough data. We don't know enough about the side effects yet or the real efficacy. But I do find it curious that they use the term vaccine, since a vaccine is typically used before you get a disease, and this is obviously after someone has contracted the brain cancer. So Andy and then Chad, what do you think about this study and its possibilities for the future?
Well, I mean, it's hard not to get excited, and I'll probably even connect the dots from a few other things that we've chatted about. I mean, one, just the unmet nature of this devastating disease. Anything that's a possible solution, even if it's a bit premature, I don't think it's premature to get excited about it, because again, we desperately need something that from a therapeutic intervention standpoint that can help manage this disease. And so I think even in the early stages, I think that's it's okay to get excited about it. There's nothing wrong with that at all. I mean, this is an area where some of the most excited I've been as a scientist is even way back in lead optimization, before you've kind of declared a candidate. You've just seen, you kind of start to, maybe you get a little Pollyannic, but you start to just wonder just how, what the potential of this product could be in terms of alleviating human suffering and transforming human disease. So it's, so I'm excited even just by that headliner. Yeah. I think, and I'm kind of therapeutic agnostic, so I don't like small molecule, large molecule, cell and gene therapy. Whatever provides the best solution to the patient is what I would care about. So again, I'm not one of those that is an anti-vax and saying like, oh no. Not at all. I mean, I think if this, again, whatever provides potential benefit, I think we also talked a fair bit about risk benefit. So in this situation, I mean, this is kind of the one extreme of risk benefit. I mean, it's a disease where it's almost all risk, and anything that comes along with benefit, even if it has a little bit of baggage, is probably going to be evaluated very, 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 very focused, and probably with even a little bit of regulatory flexibility to give this product every chance to be successful, to have an impact on those that would otherwise have maybe no therapeutic option. Now I may have taken this a little bit too literally, but did you actually even indicate in the headline that there was actually heat, kind of
well, yeah.
Or
so that's a
hot
terminology.
topic.
So the idea, right? No,
well,
from cold
I
to
think
hot.
I think they're calling it hot because they're like activating it, right? Okay. So it's like going from cold, like inaccessible, to activating it, and yeah. So that's where the hot came in, and yeah. So Greg, you had brought up the part about the vaccine. And it's a vaccine because you're dosing, you're teaching the body to fight it itself. And what was interesting here also that I don't think you touched on in your summary there, but they called this kind of like an onion, and then they went on to say it's actually even like a bag of onions. And one of the challenges with a lot of the immunotherapies right now is they don't stimulate the immune system as much as they would like to. And what I'm inferring is that this the onion is kind of this delivery, but then the bag of onions is this aggregation of delivery vehicles. And when you're thinking of dosing large molecules, one of the things you want to stay away from is aggregates, right? And I'm reaching here and I'm kind of guessing, but aggregates are bad usually because they stimulate the immune system. But I'm thinking here, this aggregation. So when they talk in this article about the bag of onions, they're delivering this aggregate. So I think it's actually stimulating the immune system to make this even more powerful. So they've kind of wrapped a few different things into this in a really clever way with some fun terminology. For
sure. And before Greg goes, the other thing that I was probably most, again, the preclinical scientists in me, again, using dogs, which have kind of a spontaneous disease, which I would say is probably very pathophysiologically similar to the human disease, I'm assuming. So even that's a wonderful model in terms of, to also put your hope into in terms of its potential translational the predictive utility of that in terms of predicting clinical outcome. I've often thought, again, a lot of the animal models that we have are fairly contrived. They're kind of genetically manipulated, chemically induced. They're a bit artificial. But I've often thought, again, in particularly large vet hospitals, which would have access to not animal models of disease, but animals that have spontaneous disease, which would be much more analogous to the clinical disease, and then evaluating the potential therapeutic utility in those models, which was done here. I start to get even more excited about the predictive utility that might have on the clinical outcome. So, and I know even a lot of big pharma has thought about maybe establishing deeper collaborations with large colleges of veterinary medicine to probe that exact opportunity, that exact topic. So,
yeah.